The three major zones, from inside to outside. We still use the three zones, but we'll explain the layers sequentially, moving from the inside of the eye, where light hits first, toward the back, where the signal starts. 1. The output and wiring zone, layers 1 to 4. These layers are closest to the vitreous humor, the jelly filling the eyeball. Their job is to prepare and send the final signal. Inner limiting membrane, ILM, this is the smooth, innermost, glass sheet that separates the retina from the vitreous fluid. It's the floor of your eye's inner cavity. Nerve fiber layer, NFL, this is the highway of wires. It consists of the bundled axons, output cables, of the ganglion cells. All these wires run across the retina and eventually converge to form the optic nerve, which leaves the eye. Ganglion cell layer, GCL, this is the CO's office. It contains the cell bodies of the ganglion cells, the final output neurons. They decide if the signal is strong enough to send to the brain. Inner plexiform layer, IPL, think of this as the final handshake station. It's where the middle manager's bipolar cells, synapse, connect with the CEO's ganglion cells to hand over the processed data. 2. The processing and cell body zone, layers 5 to 7. These are the middle floors where the bulk of signal processing happens. Inner nuclear layer, INL, this is the middle management floor. It holds the cell bodies of the three main types of processing neurons: bipolar cells, the direct signal path, and amacrine and horizontal cells, which refine contrast, edges, and motion. Outer plexiform layer, OPL, this is the raw data input station. It's where the photoreceptors, the sensors, synapse with the bipolar and horizontal cells. This is the first stop for the light-generated signal. Outer nuclear layer, ONL, this is the sensors control room. It contains the cell bodies, nuclei, of the rods and cones, the photoreceptors. 3. The sensory and support zone, layers 8 to 10. These are the outermost layers, where light is actually captured and supported. External limiting membrane, ELM, this acts as a support scaffolding. It's not a true membrane but a series of tight junctions that mechanically hold the photoreceptors in place, acting as a filter or barrier. Photoreceptor layer, PRL, this is the light sensor array. This layer contains the outer segments of the rods for dim light, and cones for color and sharp detail. This is the crucial layer where light energy is converted into a chemical, electrical signal transduction. Retinal pigment epithelium, RPE, this is the maintenance crew and light trap. It's the outermost layer, sitting right next to the choroid blood supply. It contains pigment to absorb stray light, preventing glare, like black paint inside a camera. It provides nutrients and recycles the outer segments of the photoreceptors, which are constantly shedding. Key takeaways, the alternating rule. To keep it simple, focus on the alternating pattern in the middle. Plexiform layers, IPL, OPL, equals wiring, synapses, where cells connect. Nuclear layers, GCL, INL, ONL, equals cell bodies, where the brain, nucleus of the cell is. This organization ensures that the light passes through the processing layers first before hitting the sensors, PRL, at the back, and the resulting signal travels back through those same layers to exit via the NFL optic nerve.